little cozier. Great. There we go. Much nicer. Friendly. Honestly, it's if you're someone giving a talk and like there's like these rows of empty seats and everyone clustered to the back, it's just a different experience to actually be able to see the faces of the people you're addressing. Great. Well, good morning, everyone. We have a special Grand Rounds today, and it's quite a wonderful special Grand Rounds. Our speaker today is Brian Sweis, who many of you know. He has been in our graduate program in neuroscience and is now a medical student in our medical school. And what's been exciting about Brian's career thus far is his immense creativity and productivity. And I want to point out that he is still a medical student. He has not graduated. I want you to look at this CV here. This is a CV of a junior faculty member, just in terms of his publications, his presentations, the innovation of the work he's been doing. It's really, truly remarkable for someone at his stage in his career. The other thing that's very kind of exciting about Brian's trajectory is that he has a longstanding interest in neuroscience. He got his bachelor's in psychology and in biology with minors in neuroscience and philosophy from Loyola University in Chicago. But with that background, and he entered graduate program here in neuroscience, he found himself, as he was delving now into some of the areas of studying decision-making in the brain, which was his area of interest, he found himself a little bit torn between neurology as his future career in terms of the medical component of what he would do versus psychiatry. And I, of course, did my best to persuade him in one direction or the other, but knowing that ultimately he would of course be making up his own mind. And earlier this academic year, Brian did his neurology rotation, followed by his psychiatry rotation. And as he just said to me, sitting here next to me, the psychiatry was just a lot more fun. And I think it's not just the fun component, but as he said to me in other conversations, the clinical issues and the clinical problems that are facing psychiatry are of a depth and a complexity that really are going to require these latest and newest tools in terms of dissecting what is it that goes wrong in these higher order neural systems, these higher order behavioral output systems in the brain. That's the only way we're going to be able to start to really understand the complexity of psychiatric presentations. And Brian is especially well poised to do that work. I've mentioned his background in education. I will just mention briefly in passing the overall thrust of his work, because you're going to be hearing about it here. But it is to do exactly that. It's to take the current tools of experimental neuroscience and to join those tools with approaches that are from computational neuroscience and moving in that direction of computational psychiatry, but also joining those tools with the emerging field of neuroeconomics, of how human beings make decisions, and putting that all together in a very novel framework to help us understand when faced with multiple different kinds of choices that might lead to multiple different outcomes, what are the decision-making systems that are called into play that push into one set of decisions versus another, and how might those go awry in ways that are relevant to psychiatric illnesses and to addictions. So thank you very much, Brian. Looking forward to your talk.
Can you guys hear me okay? Is that all right? Thank you, Sophia, for that introduction. That basically is the entirety of my talk. Probably don't even need to give a talk at this point. She drove the message home. So however I end, depending on how much time we have, that's essentially the message I want to leave you all with. And I just kind of want to start to begin with thanking my two PhD advisors, David Reddish and Mark Thomas. If I don't thank them now, basically every message in all of the slides throughout the rest of my talk, they played a tremendous part of that. So to start, I have 3 learning objectives that I want to get across. It's this concept of neuroeconomics, which I'll just read off here. It's this idea of characterizing unique decision algorithms or mental calculations that we use. and how information is accessed and processed differently depending on distinct economic theories that might be driving those types of actions. The other component I want to touch on is this concept of computational psychiatry. That can we evaluate psychiatric disorders or the interventions we choose, not by symptoms alone, but by the computational process that's dysfunctional in that individual. And the third thing is this idea of plasticity. I feel like that's a buzzword that gets used a lot, and I really want to make it concrete and operationalize plasticity, or specifically synaptic memories, in an objectively measurable way that we can quantify and manipulate experimentally. So to get into this, I want to talk about this concept of computational psychiatry, and David Reddish and Josh Gordon, the new director of the NIMH, essentially put this message out there that before we can treat any disorder, one must know what the disorder is to begin with. And I use this analogy with chest pain when, as medical students, we are trained to think of a differential diagnosis of all the things that could underlie chest pain. There's a whole number of fundamentally distinct pathological mechanisms that could contribute to chest pain, each of which have their own laboratory diagnostic workup, and they have their own fundamentally different intervention. So mental health, unlike other fields, generally uses symptoms as the sole basis for diagnosis, for intervention selection, or even monitoring how treatment progress is going. And what I want to get across is, can we think of biomarkers of pathophysiology, or even compensatory mechanisms we can exploit or take advantage of, or even just measure behaviors to get at something more than behavioral symptomology that's rather generic? And can we tailor each of these to an individual's neural process that's gone awry? So with that in mind, I want to use addiction as an example of this. And when we think of all individuals who use drugs, the first thing we know is that not everyone goes on to develop problematic drug use, which we have to think about when we study addiction in animal laboratory models. Of those who have problematic drug use, is there actually something going wrong inside that individual? Or is it solely due to their environment? And that if you change their environment, the problematic behaviors go away. As opposed to also within an individual, is there something wrong with neural changes in the brain? Or are there non-neuronal issues going on? And there's a lot of research suggesting that glia and other key players in an individual's biology give rise to some of these pathological behaviors. And of those neuronal changes, how many of those are normal mechanisms of learning? Addiction is thought to be a disorder of learning and memory because drugs of abuse can give rise to lasting changes in the function of the brain. And is that something that the brain would normally do, or is it a breakdown of learning mechanisms? And finally, we like to jump all the way from addiction to dopamine issues. And there are different mechanisms in the brain that could give rise to changes in synaptic memory that don't have anything to do with dopamine. And so I use this cartoon of a rodent brain just to illustrate one neural system that where these blue neurons here represent a subset of dopamine neurons projecting to all different parts of the brain. And so if you choose any pathological mechanism that you're interested in studying, then the question becomes, where in the brain are each of these things malfunctioning? Is there a dopamine issue here, or is there some other issue related elsewhere? And how might those changes give rise to the way the brain processes information in a maladaptive way? So with that in mind, I want to just start with a very simple rodent task and an odd finding that has been going on in our lab. And so one simple animal model of addiction-related behaviors, specifically studying addiction as a disorder of learning and memory, is this model of a conditioned place preference behavior that you can easily measure in animals. It's a very simple experiment. You put an animal in a 2-chamber box and let the animal just run around. And over time, you can just quantify at baseline how the animal chooses to freely explore this environment, where the colors here show how the animal spends 10 minutes in this box. Then what you can simply do is you can pair an animal in one chamber, lock it in this one room, and inject the animal with a substance of abuse that you want to study, like morphine. And on the other side, on a different day, you can inject the animal with saline. And if you repeat this over and over again, you're conditioning the animal that this location is associated with a particular drug of abuse. Then you put the animal back in this free exploration chamber and see where they spend their time. Very simple behavior. And what you can see here is this animal has been conditioned to spend most of its time with the drug-paired chamber. Then you can continue to put the animal through a relatively simple additional learning paradigm where you just put the animal in this box, let it freely explore over and over again. You can repeat this for a week and see where the animal chooses to spend its time. And you can watch this behavior extinguish over time, as if the animal is forgetting or no longer interested in that particular side of the chamber. This has been used to study addiction in the animal laboratory model, where most of our understanding of the biological mechanisms of addiction come from what is going on at each of these time points in the brain. And if we want to study relapse, or at least chase a biomarker of relapse, you can reinstate this initial behavior. And the ways you can do this in an animal model is present the animal with different triggers. You can inject the animal with drug again to remind the animal what that feeling of having drug on board is like. You can play a tone that you may have associated with the initial associations, or you can stress the animal out, and all of these things have been shown to reinstate this drug-associated condition-place preference, as if the animal is retrieving some earlier memory of that behavior. So coming into the laboratory of Mark Thomas, we can do things to the animal, and I'll get into the details later, but thinking about new tools to study circuit dysfunctions, can we do something like deep brain stimulation and pick a target, a circuit of interest that you want to study, and I'll get into this later, how we can specifically target individual pathways. And can you do something to the animal that prior to giving the animal a trigger of this relapse-like behavior, can you block that reinstatement behavior? And this was published by a former postdoc in the Thomas lab, that there's certain circuits and different changes you can do to plasticity in certain pathways that can block reinstatement, right? And so this might seem like a potential treatment or therapeutic option to prevent future relapse. If you repeat this exact same experiment, except you change the drug of abuse, and you expose the animal to cocaine instead, and you do that exact same neural manipulation that is therapeutic for an animal treated with morphine, what we found was that stimulation alone caused the animal to relapse. Right? And this becomes a very peculiar finding when you think about different treatment options, or on the one hand might be therapeutic for one individual, and that exact same treatment intervention by itself pushes the individual over the edge. And so we have to really think about what changes are we inducing in the brain, and what does that mean in the broader context of what that brain is normally doing when choosing to do one thing over another. And that not all treatments can be tailored to different individuals the same way. So to think about how do you explain the action selection process gone awry, other individuals have tried to study in this simple paradigm, what is the animal doing when it's transitioning from the morphine-paired room to the saline-paired room, or vice versa. And I'm not going to get into the details of it, but you can start to ask questions like, let's put electrodes into the brain of the animal and listen to brain activity right when the animal is about to leave the morphine room, or vice versa. And there might be certain changes that change in these animals as a function of their history of drugs of abuse. And this starts to get at this question, this action selection process, what's actually malfunctioning every time a decision is being made. But you can ask yourself, when this animal is leaving the location paired with the morphine side drug, is the animal asking itself, is it worth leaving this location? And you can compare that against a different type of decision. Might the animal be deciding between, is the morphine side room and the saline side room better or worse? And those actually are fundamentally different questions and different algorithms in the brain used to calculate the action about to be selected. And so I want to get into behavioral economics a little bit, where if you look at any action that's being selected, you can say that as a function of cost, every individual's value of choosing an action will decrease. The more expensive something is, the less likely you are to do it. And so you can quantify the slope of this discounting function by measuring this particular discounting function. And then you have to ask yourself, what is an individual's threshold before they no longer do something, right? And so anything on the cost axis that's less expensive than this particular threshold might be the tipping point for an individual to choose an action. And anything to the right of this threshold on this cost function is when the individual would say it's no longer worth choosing that action. And the question becomes, how do you define that threshold for different individuals, depending on the type of question that you might be asking internally? And there might be fundamentally different economic theories that drive these behaviors. And I'll get into this later, but I want to briefly mention, are you choosing a particular action as a function of this threshold that's determined by some average reward expectancy in the environment? That if something becomes too costly, that's how you make a decision, and this value is set. It's a set fixed number. As opposed to a different type of decision where you're choosing you're making a decision to choose option A over option B, both of which follow this discounting function, and you're now choosing and weighing the options against one another. Whereas this type of behavior, this is it worth it behavior, represents a fundamentally different decision than choosing between two options. Right? I also think of this as you're either opting in or opting out of what you're currently doing. So these tasks, whether it's an animal learning to go to one side of the chamber, or even in other simple tasks where the animals are pushing buttons to get infusions of drugs of abuse, really these tasks are designed to study mechanisms of learning and memory, where the interesting things that these studies are really looking at is what happens between these different periods of learning a behavior, extinguishing a behavior, and reinstating that behavior. All of these get you closer to understanding mechanisms of memory, when a memory is extinguished versus when a memory is reinstated. And what I'm interested in is understanding the decision process that you're choosing every step along the way, right? So if you're a mouse deciding which room to go into, what are the mental calculations that are taking place? And even if you're a mouse that's reinstated behavior, what are also those mental calculations you're doing that it might be different in one state versus the other? And how can we use decision-making to study the action selection processes that are changing, not just the memories that might be changing? So to get at this a little bit further, I want to move towards, instead of these simpler traditional paradigms, different paradigms that move us on the scale of learning and memory to how we're accessing information in the moment. And to do this, I move into this realm of neuroeconomics. And what this can really tell us is more complex components of information in the environment that might give rise to how behaviors influence one way or another. And really what this is doing is it's moving closer and closer to a faster time scale of understanding what's happening in the moment, as opposed to what's happening across days. So moving forward, trying to think of animal behaviors that can become more complex to model more complicated forms of decision-making, I'm moving into a different decision-making paradigm that essentially started when I came into the laboratory of David Reddish. It's a very simple animal task. You drop an animal in this maze where it can just run around a square maze foraging for food. You take a hungry mouse, put it in this maze, and he'll run around naturalistically looking for food. You can put different flavors in the corners to encourage the animal to go to different corners to earn different pieces of food. But essentially, you keep the rules pretty simple. You give the animal an hour to earn all the food that it can for the entire day, and you don't feed it otherwise. And that gives the animal motivation to run around this maze looking for food. And we don't have to assume anything about which corner is more valuable from one animal to the next, as every animal might care about different flavors to different degrees. And this was the work of Adam Steiner, who created this task in the Reddish lab. And what I did was started to make the rules of this task a little more complicated. But you can look to see how the animal spends their 1 hour budget, because every decision the animal makes is at the expense of doing something else. And so now time is money on this task. And if we make it a little bit more complicated and add some extra rules into the mix, we can start to study some of these decision-making processes in mice. So I'll keep this pretty simple. When the animal gets to a fork in the road, we can play a tone, and the tone can tell the animal that food is on its way. And we can train the animal on a couple extra things. The pitch of the tone can tell the animal how long it will be before the animal gets its food. The higher the pitch, the longer the wait. And we can see now, when pressured on a limited budget, how these animals make these economic decisions. So the first tone the animal will hear is a 5-second tone, and the animal will accept it, and then have to wait 5 seconds in this waiting chamber before it gets its piece of food. Right. A little piece of food shoots out in the chocolate station, the animal eats it and moves on to the next station, which we give a more expensive offer in this case, and the animal decides it's not worth it. Right. So essentially, this animal is running around a food court. It has a 1 hour on its lunch break to choose to spend what it decides is worth investing in versus what might not be worth investing in. And in this next example, we'll give the animal a 16-second offer for a plain flavored pellet. And you'll notice that the animal hesitates quite a bit before accepting that offer, and then accepts it. And the animal doesn't have to stay there. At any point, the animal can change its mind and decide, you know what, screw it, it's not worth it. I'm going to change my mind. And we can look to see how the animal spends its limited 1 hour budget, choosing between all of these different actions. So what I show you here are these different behaviors that the animals could be competing between, represented in these different colors, where green is an enter decision and red is a skip decision, versus pink is this change of mind decision. And we can just watch how the animal spends its budget over the course of an hour to really understand how every action is selected among a bunch of different competing costs that the animal might be choosing between. So we watch these behaviors, and just to show you a rough estimate of how an animal spends its 1 hour budget, animal spends a bit, maybe a third or a quarter of the time choosing between entering and skipping, maybe another quarter of the time choosing between skipping and actually earning the piece of food. And then what we noticed strangely is an animal spent nearly half of its 1 hour budget after it earned a piece of food before it moved on to the next trial, which was bizarre. Right? It was a really bizarre fact that the animal spends 30 minutes of that limited budget. And if we split this across the 4 different restaurants that the animal is choosing between, where the first ranking restaurant is the restaurant the animal earned the most food in, we can look at behaviors as a function of subjective value. So the first thing you notice is that this cyan-colored line, which is the time spent earning food, obviously is the highest in the restaurant the animal cared the most about, which makes sense. They're most willing to spend time earning that food. But other interesting bizarre behaviors come up, like the time spent after consuming food before moving on to the next trial was also the highest in their favorite restaurant. And we tried to figure out what's going on with that sort of behavior. We even put little cameras in the animal's room to see what they were doing, and they essentially gobbled down the food instantly and then just sat there for an extra 10 seconds, wasting time, and they did it longer in the restaurants they cared about. If you look at some other behaviors, like the time it spent for the animal to say no, that behavior got longest for the restaurant the animal cared the most about, which is also bizarre. And I'm going to dive into this a little bit more about what this means. But the nice thing about a neuroeconomic task is I can spend a week at a time changing the environmental richness and scarcity of the reward availability in the animal's environment. And so when I actually look from a reward-rich environment early on versus a reward-scarce environment where offers can range from 1 to 30 seconds, that's when we see that decision to say no take the longest. Right? When things become scarce and the animal has to walk away from a potential reward, that decision becomes tougher. And if you actually split that up by these animals' preferences, that decision becomes toughest for its most preferred restaurant, and that they learn this behavior over the course of 2 months or so. And if you look at the different behaviors, like the decision for the animal to change its mind, that too is toughest in the animal's most preferred flavor, but it learns on a different time scale. Right? Like the decision to say no and walk away from a particular offer might be a completely different learning process than the decision to change your mind, and that these operate on completely different timescales. So what we can do is we can break down these trials, trial by trial, looking at these different offers these animals are exposed to, where if you look at these example trials and you ask did the animal accept or reject these offers, we can start to quantify and crunch the numbers of the economics of these behaviors. And if you look at the offers that the animals accepted, you can see which ones they earned versus which ones they changed their minds on. And if I plot this data, particularly for this initial offer zone, where they're deciding to choose or accept or reject an offer, we can start to quantify data in a way where if you fit this to a logistic regression, we can figure out what is the animal's willingness to accept or reject an offer. And for this particular animal, it's around 12 seconds or so. Anything above that, they generally said no to. Then we can also look at the second decision they make, which is their decision to change their mind. And we can quantify that particular willingness to actually stay there and earn their food. And we can quantify that with a different threshold, where even though the animal is willing to accept up to about 13 seconds, it's actually willing to stay committed to offers only at around 10 seconds or so. Right? And so now we can start to quantify in these different flavor preferences how these animals make these different types of decisions. And what we immediately start to see is in restaurants these animals care the most about, like banana for this animal, you see a gap between what they're willing to choose between in the offer zone and what they're willing to change their mind about. Where for restaurants these animals don't really care about, the decisions they choose to accept or reject are the decisions that they often end up remaining committed to. And so this opens up an interesting window, particularly for these types of decisions, where now we can operationalize in an animal what they should versus shouldn't do. And really in animal behavioral studies, you can't really impose on what a mouse ought to do. And using an economic paradigm, we can quantify that. So in this scenario, if an animal should skip, that's the valid thing to do. However, if the animal were to enter that decision, that's an economic violation of their own subjective policy that we can watch over the course of 100 straight days when we test these animals in the laboratory. And so what this behavior looks like, if you look at this valid decision to skip and the valid decision to enter versus this economic violation to take something they shouldn't do, this is what the behaviors look like. These animals run right in to accept an offer they very well should take. And if you look at the decisions to say no, these animals actually catch themselves before they walk in, and then they say no thank you. Right? And this is why those decisions to skip take a little bit longer, is they seem to be catching a mistake about to happen. Sometimes, though, that mechanism, whatever is going on here, isn't engaged. And when these animals make economic violations, they tend to just run right in. And actually, that behavior looks identical to the decision when they should take those offers. And so we can quantify this behavior, but then we move into a different study where we now wanted to see what might the effects of different drugs of abuse be on these types of behaviors that we can operationalize on what is economically the valid or invalid thing to do. And so just to show you what I showed you a second ago, the decision to skip took a lot longer than the decision to enter, regardless if it was the valid or invalid decision to make. And if you expose animals chronically to saline over the course of time, these behaviors don't really change. But now if I expose animals to a week of either cocaine or morphine exposure, and then look at their behaviors 2 weeks after their last dose of drug, this is long after drug has long cleared their system, at a time point where we know certain circuits in the brain are remodeling. What we see something is interesting and different between these two drugs of abuse. What we see is for these cocaine animals, only the decision to choose to accept something that they should not be accepting in the first place gets a lot slower. And I'll come back to this in a second, but behaviors in these morphine animals didn't change at all. If you look at the second stage of decision making, where these animals could reevaluate and change their mind, we can also quantify how valid of a decision is that. And what I can show you is the majority of these change of mind decisions are actually the economically sound thing to do. And what that means is, and I've demonstrated in this graph, is that if an animal accepted an offer that they really shouldn't be taking in the first place, and you watch to see as they're waiting for it, the moment they quit is actually before they might as well just stay there, right? As if this was an offer that they should have skipped in the first place, right? And so the majority of these quit events are actually economically smart. It would actually be very foolish for this animal to change their mind at the last moment. And so what we found interesting on the morphine side of things is their threshold to change their mind increased. And what that looks like on behavior is that blue line slides to the right, where these animals aren't changing their mind to correct an economic mistake that they made. The way they made the mistake in the first place looks just like every other animal, but their capacity to correct a past mistake was impaired. So if you compare these two different animals side by side together, what the cocaine animals are actually doing in the offer zone is they're about to walk away from these poor decisions, which is actually the right thing to do, and then spend time thinking about it to ultimately accept them anyway. And these morphine animals, when they do make a mistake, they kind of run right in. And when given the opportunity to fix those mistakes, they elect not to. Right? And here now we have two completely different forms of impairments in decision-making that separate these cocaine and morphine animals that you might not be able to separate on a simple behavioral paradigm. And I'm not going to get too much into the details of it, but now you have two different types of decision-making vulnerabilities where should you do the right thing versus should you do something that might not be in your best interest, where that process might be malfunctioning in one type of subset of patients, whereas in a completely different subset of patients, after you've made a mistake and you have an opportunity to correct that behavior, could that be another decision-making vulnerability that could live in a completely different circuit in the brain? So Sheila Specker, I was working on an abstract with her, and she crystallized this idea in a simple way based on the human narratives of the patients that you see in the hospital. Individuals who desire to recover from addiction often make poor decisions and relapse despite this desire. And what we think we're getting at is we're operationalizing, even in animal behavior, a crystallized form of an internal conflict between what you know you might want to do on the one hand versus what you end up doing that might not be in your best interest. And what I'm showing you here is we can operationalize that in simple animal behaviors in completely different types of conflict. I'm not going to get too much into this, but the rest of the Reddish lab, and David's finger is pointing it out in this animation, is spent on understanding the neural processes going on when you're making these decisions, where parts of the brain might be thinking about different locations as you're trying to choose between actions. That's the rest of the Reddish lab's bread and butter, and I'm not going to get too much into it, but there's a whole body of literature that explores what's going on in the brain during each of these moments. So I want to return back to this concept of economic theories of decision-making, where in a deliberative model, if you're choosing between option A and option B, that's what we think is underlying the threshold in the offer zone when animals are making skip versus enter decisions. And I want to play a thought experiment just to really get this point across. So let's imagine that you have a bunch of different offers that could be on the plate. And on this particular trial, let's say you've encountered a 1-second offer. Okay? And if you're trying to choose between what you should or shouldn't do on this particular trial, let's say you pause and you imagine that, well, around the corner, there might be another 1-second offer. And so if you're asking yourself to make a decision, you might be pretty unsure on what to do if these are equally cheap, right? And that's indicated by this green dot. If the current offer gets more and more expensive and you're still imagining that there might be something better around the corner, as a function of that cost, you're definitely going to skip this current offer and you're going to go chase that other offer around the corner. If I change what's going on in your head and actually you're imagining that actually there might be something really bad around the corner and the current offer is 30 seconds, you still might be equally unsure on what to do. And if the current offer comparatively gets cheaper than what you imagine is around the corner, you're definitely going to stay and take this deal. And so what I just illustrated here is this decision playground that you have when you're imagining what the future might hold, right? And it's this three-dimensional space where that green space is this area where you're not really sure what to do as a function of all the costs you could encounter and all the possible things that might be around the corner. This is what I mean by a deliberative model, and you can model this with this function. If I go back to the classic addiction literature, where they look at individuals with addiction and their discounting functions, the general consensus in the community is that individuals struggling with substance abuse have a steeper discounting function. This is what we mean by impulsivity, is that your value of doing something as a function of delay rapidly drops off. And that delayed gratification is difficult for individuals with substance abuse to wrestle with. In a deliberative model, if you model this increase in this discounting function, this K parameter, it actually doesn't change your threshold on how you choose between decisions in the offer zone. But what this looks like on this three-dimensional plane is if you increase this discounting function, your plane actually flattens out and there's more of this unknown space in a deliberative model. So really, when I use the word impulsivity to describe behaviors of an individual, and you think about it in economic terms, it means a whole lot more. It's a pretty loaded statement. And what we think is happening with these cocaine-treated animals is in the offer zone, while they're not changing how they make decisions as a function of cost, how they deliberate, particularly when they're faced with offers that might be good or bad, they're really shooting in the dark. And they're making decisions with a lot of unknown territory about what happens in the future. And we think something completely different is happening with these morphine animals. In a foraging model, if we model this increase of K, just like the rest of the addiction literature says, what that actually does to this weight zone threshold is it pulls it to the left. And that's not what we see in these morphine-treated animals. Also because they don't show any changes in the offer zone when they're deliberating. But the other term in this foraging model is this R term, this expected reward in the environment. And if after having experienced a drug like morphine, I think this aligns a lot with the clinical anecdotes of patients where after having experienced a drug of abuse like an opioid, that everything else pales in comparison. And what you can model in this particular framework is a decrease in just what else is out there in the rest of the environment. And that everything else sucks in comparison. And so your average value of reward in the environment shrinks as a result. And what that does to decisions in the offer zone, it actually pushes the threshold to the right. And it could actually change your ability to have a change of mind decision. That if what you're currently doing compared to what else in the environment is probably better and everything else out there sucks, you might be less reluctant to fix a previous mistake. Right? And I wanted to spend time there because we often talk about this chocolate cake hypothesis, that in addiction, is there anything different between a drug of abuse and really, really good chocolate cake? Right? Do they lie in the same value continuum? Or is there something fundamentally different about drugs of abuse? And what I'm trying to illustrate here is there's completely different decision-making processes that are malfunctioning that might not just have to do with pleasure. So this is one of our papers in Nature Communications, and I think for the rest of the talk I want to move a little bit faster to get into the neuroscience behind how we can start to dissect some of these processes. So when thinking about different traditional paradigms and different economic paradigms, and now newer tools of neuromodulation, I think of two ways of manipulating brain activity. You could do it online while the animal is behaving. So in these simpler paradigms, that could be stimulating the animal during this conditioned place preference paradigm. Or even in a neuroeconomic paradigm like my task, where you can stimulate the animal during decision making. And these are experiments that we can certainly do. These online neuromodulation approaches can sometimes be referred to as continuous stimulation, stimulus-locked event-related, closed loop, or on-demand, are a lot of buzzwords now to make neuromodulation smarter, tied to different things in the environment. But I want to focus on this offline form of neuromodulation, where let's manipulate particular pathways outside of the behavior we want to study. And then let's set the animal back into their natural environment and see if we can augment behaviors long-term. Right? And that can be true if you're using a simple paradigm or a complicated neuroeconomic paradigm. And this is the approach that I took for the rest of my dissertation, is I want to choose a task that is information access oriented, but I want to choose a manipulation that can manipulate storage properties or plasticity. If we think addiction is a disorder of learning and memory, let's manipulate that mechanism, but use a task that can tell me something about different competing valuation processes in the brain. So how do we gain access to a very specific circuit, like this particular pathway? And with mouse tools, we have a way to do it. So this is a particular tool called optogenetics. And essentially what we do is we can engineer neurons in a particular spot of the mouse's brain to manipulate just that pathway and nothing else. And the way we do this is we put an animal under surgery, brain surgery, and we can pump a particular virus into an area of the brain we want to study. And in brief, what this virus does is it introduces DNA into a neuron. And that DNA encodes a protein that can be expressed on the surface of cells. And this protein is an ion channel, but what's unique about it is it's a light-sensitive ion channel. So if you shine a beam of light or a laser beam on the surface of this neuron, you can now control action potentials of that neuron. Right? And because this is all under genetic controls, we can specify what particular cell types we want to manipulate. The other really neat thing about this is just to illustrate that ions can flow into this channel only when I, as an experimenter, shine light on that particular neuron. What we can do is now decide how do we get light into the brain of the animal. And if you've ever had one of these Christmas trees before, these fiber optic Christmas trees, we essentially just break off little pieces of those and make implantable versions of those flashlights. And then we pick where we want to surgically implant those into the brain of the animal. And now an awake behaving animal can receive light delivered to a very specific spot of the brain. The really neat thing about this is that if I infected neurons up in the prefrontal cortex of the animal, specifically the infralimbic nucleus of the prefrontal cortex, those neurons all have these special light-sensitive ion channels on them wherever they project to. So if I shine a beam of light down into the nucleus accumbens, instead of just electrically zapping any neuron that's in this part of the brain, I will specifically activate action potentials in just those neurons and release glutamate or whatever our neurotransmitter of interest is. Right? So now I can very specifically manipulate a certain set of synapses that I want to study. So I'll just fast forward through this just to show you that down in the nucleus accumbens, the neurons that we've genetically altered all fluoresce green here, and these blue cell bodies are the cells that live in the nucleus accumbens. And so there's a big bundle of axons coming from the prefrontal cortex that I have access to very specifically manipulate. So the nice thing about working in the Thomas lab is they're experts in how do you actually take the brain out of the animal and keep it alive in a petri dish with artificial CSF and the right nutrients it needs to stay alive for a day. And if you cut thin little sections, where I'm circling the nucleus accumbens in pink here, I can put that under a microscope, keep that tissue alive, and record from these synapses that I want to manipulate. And so what this looks like under the microscope is I shove a recording electrode into the nucleus accumbens, I put my optic fiber over this tissue, the tissue's still alive, and if I shine a beam of light onto this tissue, I can record this electric waveform. And I'm just going to name this first peak, this N1 component, and the second peak, this N2 component. It's the second negative peak. And just to illustrate what these waveform components are, if I put in a recording electrode here and I shine light onto the terminals of neurons coming from the prefrontal cortex, over the time course, the first thing that happens is we get an action potential here, neurotransmitters are released, and then you get a postsynaptic response. And so we know this first peak is the presynaptic component coming from the prefrontal cortex, and the second peak is the postsynaptic response. And we have the pharmacological experiments to test that and prove that. This is work that was helped tremendously by Dr. Aaron Larson and Manny Esquera in the Thomas Lab, that I couldn't have learned any of these techniques without them. And so now I want to get into work about how do we manipulate the specific memory of this circuit. So what I want to do is essentially measure what these waveforms look like before and after an experimental manipulation that can rewire the circuit. And ways we can do that with stimulation is you can stimulate parts of the circuit with rapid tetanus 10 hertz or 100 hertz stimulation parameters to rewire the strength of the circuit. And what this looks like is if I just stimulate this particular system just once a minute just to see what this looks like over the course of an hour, I can see that just by doing nothing over the course of an hour, these waveforms don't change at all. If, on the other hand, I stimulate this tissue with a 10 hertz tetanus for 10 straight minutes, and then I go back to looking at what this system looks like, I notice that I get a significant reduction in the second peak without affecting the first peak, and that this lasts for at least 45 minutes. What this looks like, and what I think is happening, is this presynaptic neuron and this postsynaptic neuron, how they communicate with each other, is weakening, right? I'm not changing activity in the presynaptic neuron, but how that postsynaptic neuron receives information is significantly reduced. And this is one way to measure synaptic memory, and I can artificially manipulate and weaken this connection. If I do the same procedure with 100 hertz stimulation, there's no effect on the strength of the connection between these two sets of neurons. So this now opens the bread and butter for the rest of the Thomas Lab that has really become an expert in understanding how mechanisms of plasticity can change in the brain. So what I also wanted to do was I wanted to develop a way of not just knowing if I could change the strength of a network, but could I actually measure how strong one mouse's infralimbic to accumbens connection is compared to a different mouse. And I developed this new method to do it. So if I stimulate this tissue, and I record my N1 peak and my N2 peak, and I just plot this on a graph against one another, right, where that data point shows me my N1 peak and my N2 peak. And then I just crank up my stimulation, and I do this more and more, and I get stronger and stronger response out of these two sets of the presynaptic and postsynaptic neuron. Something that we wanted to figure out is there a way to measure how this circuit is tuned? And the way I tried really hard to think of an analogy to get this method across. And if you've ever had one of these tuning devices and you've ever tuned a guitar or a piano, and you run an instrument through the scale, like this, essentially that's what I'm doing here, is I'm running this circuit, this synapse, through this scale. And what I think we can use is the slope of how they respond to this range of intensities as a metric of how strong the synapse is. And the other analogy I thought about was if this is a presynaptic neuron in dollars and the postsynaptic neuron in euros, that this slope is the exchange rate of this currency, right? Where now for every 2.7 euros to the dollar is essentially how this circuit is communicating, right? And that's the trade-off, or how much the signal is being amplified by the postsynaptic neuron. So then what I did was I delivered 10 hertz stimulation or 100 hertz stimulation to animals, and then a day later took their brains out and ran it through this input-output tuning curve. And what I found was this whole group of animals that received 10 hertz had a significantly less steep slope. And what that looks like on this graph is data points that look like that, where I've changed the exchange rate of neurons in this particular circuit. Okay? And really that's what's happening to those synapses, is the postsynaptic neuron isn't responding as well as it used to. So now we've created a metric where on the y-axis, and each one of these dots are individual animals, we can measure which animals had weaker synapses and which animals had stronger synapses. And I artificially weaken these connections in this group, but there might be animals that have stronger connections to begin with. And to just nail this analogy even further down, probably more than I need to, I have Celine Dion demonstrate what we think is happening to each of these sets of synapses, right? Where those might be the individual animals, and those might be animals that have stronger synapses, and these may be animals that have weaker synapses.
It's very boring.
Right? And so that's kind of this new tool we have. It's a very specific set of synapses where I can run it through this input-output curve to measure how strong this circuit is. So now going back to our neuroeconomic task, we took animals and we exposed them either to 10 hertz, 100 hertz, or no stimulation at all, outside of behavioral testing right before they went to bed, and then observed their behavior for the next week. And essentially what we found was only their thresholds in the weight zone were affected by this manipulation, not how they made decisions in the offer zone. And what this looks like on our data graph is essentially these blue lines just slid to the right, which might look familiar. All right, this is the same behavioral change that happened in these morphine-treated animals. After a week of this, we switched the assignment, and the animals that got 100 hertz now got 10 hertz. And now that group also showed a change in the weight zone thresholds. And we went back and switched the animals back to their original assignments, and did this for 5 nights in a row. And for the next 10 days, we saw again another increase in those weight zone thresholds. If I then take the brains out of all of these animals out and run them through that input-output assay to measure how strong the circuit is, what we found was that only behaviors, their weight zone thresholds correlated with how strong these circuits were. And this particular circuit didn't have any bearing on explaining how animals made decisions in the offer zone. Right? A different way to look at this is the stronger this pathway was, the lower their weight zone thresholds were, and the greater their capacity to fix a past mistake. So when you think about models or theories of addiction as being a problem of hypofrontality or weak connections in the prefrontal cortex, this really starts to break that up into different components, where the stronger the circuit was, the more likely an animal was to correct a mistake without having influence on how they made decisions about the future, which starts to tell us something different about what might be going on in the brain. So I want to bring this back to translational neuroeconomics and how we can learn from animals and bring this up to discoveries in humans. And so what we did was we took this task that was designed for use in animals, and we essentially mirrored a version that humans could perform on a computer. And while rats and mice forage for food, humans don't quite forage for food these days, but what they do forage for is entertainment on social media. And so we made a version of the task where they just surf the web, deciding to accept or reject entertaining videos of different rewards that might cost different amounts of download time. And what this task looks like is this person accepted a 3-second video to watch a cute video of kittens, right? And we can play this exact same game that these rodents can play. And afterwards, they can rate the video. And this was work pioneered by Samantha Abram with Angus MacDonald. And I learned recently that Ian actually suggested videos should be used right around the time when Vines were big. Yeah. So what we found was that all 3 species showed identical behaviors on this task. And we can start to ask questions getting at the content of irrational behaviors, right? So this is a famous study done where they asked individuals to rate how good a bottle of wine tasted. And the only thing that they showed them was the different price on the bottle, right? The secret was that it was the exact same bottle of wine. And what they ended up finding was that the more expensive the bottle was, the more they rated how pleasurable it was. Right? And so we can start to get at something like this on this task, especially in the humans, where after viewing a video, they rated them on a scale of 1 to 5 stars. And the more expensive the offer was, the higher they rated that video, right? Even if it was the same type of videos for kittens. And while I can't ask an animal how good was that piece of food, what I can look at is the exact same behavior after consuming a piece of food. And in this particular case, it was how long the animal lingered around in that restaurant after consuming a piece of food, which we know they lingered longer for in restaurants they cared more about. And the more expensive the offer was, the longer the animal was reluctant to leave, as if it's some valuation process after immediately consuming a piece of food or a reward. But this starts to think about another particular well-known human phenomenon termed the sunk cost fallacy. And I think this excerpt characterizes it pretty well. So this was a message made by this senator that said, completing the waterway project is not a waste of taxpayer dollars. Terminating this project, this late stage of development, would, however, represent a serious waste of funds already invested. The sunk cost fallacy basically captures this phenomenon that how much you pour into something actually changes how likely you are to continue to commit to it. If you've ever been in a bad relationship that you've spent so much time into and that you stay in that relationship purely because you've spent so much time is a representation of the sunk cost fallacy. Or if you ever watch a really bad Nicolas Cage movie and you're an hour and a half into it and you have to finish it just because you've already put that much time into it. these all represent economically irrational decisions because decisions should be made about the future, not necessarily about the past. That hour and a half is already gone, whether or not you enjoyed the movie. Right? And so the first thing I wanted to look at was these change of mind decisions. And the way we quantified this behavior in animals, this sunk cost fallacy behavior in animals, is I'm going to illustrate it with this example. So imagine that you've accepted a 30-second offer, and now you're in the wait zone waiting for it. Your probability of actually getting that piece of food is pretty low because you change your mind and you abandon that endeavor. If the countdown remaining was lower and lower, your probability of earning that piece of food goes up. If you accept a 10-second offer, there's probably a 50% chance you're going to finish it. If I compare a different type of trial, represented by this green dot, and the dot that I'm extending off of is 29 seconds. So if you accepted an offer that was 29 seconds, your probability of earning that is about 2 or 3%. The green dot represents that same 29 seconds left to go. The only difference is you've been sitting there for an extra second, represented by the green dot. And that drastically moved your probability of earning the reward up by 20%. And I can do this for every combination of offers. And so this green line rotated upward represents how history can influence your likelihood to remain committed to a reward. And I can do that on a continuum of different amounts of history, where the green line is 1 second of already being there, and the blue line is 5 seconds of already being there. That even bumps you up another 20%. Right? And this was the first time that we've captured the sunk cost fallacy across species, which for the longest time was thought to be unique to humans. And that this manifests itself in the wait zone during these change of mind decisions. But if I've been showing you data that suggests that how decisions made in the wait zone are fundamentally different from how decisions are made in the offer zone when deliberating, what does that mean for this irrational type of behavior that's being processed? And if you look at how individuals deliberate in the offer zone, here's the interesting thing we found, was that whether the individuals accepted an offer after thinking about it for 1 second versus after spending 5 seconds to take an offer, once in the wait zone, that did not affect their probability of earning that piece of food. Right? If the sunk cost fallacy is all about how much you've spent in the past changes how you're likely to remain committed in the future, why doesn't this history get factored into sunk costs? Right? That's kind of bizarre. And that tells me that what's going on in the brain here is fundamentally different than what's going on in the brain during these decisions. And that information about the past is being overvalued when changing your mind, but information about the past seems to be ignored when you're actually imagining the future, which is what we think the animals are doing. So I'm going to skip through some of these other parts. And essentially this was in our paper in Science, and there's a sister paper to this explaining how rats and mice in particular, are even willing to learn to avoid these sticky situations by planning ahead. But I won't get into this here. What I do want to get into is how we can use this in a translational model. And to do this, I want to have a little bit of a comment from scenes from last month's Grand Rounds from Dr. Jenny Zick, and how she brought up that when you're thinking about animal models of validity, you think of face validity, construct validity, and predictive validity. And that face validity is just, is the outward manifestations of disease mimicked in our model. And in Jenny's case, she was talking about substance-induced psychosis, whether you give a monkey or a mouse PCP and model psychosis that way. And in our case, it's giving an animal drugs of abuse, right? The animal's ingesting the drug. This is outwardly mimicking this disease. Versus construct validity, where you have pathophysiology induced with known causality of the natural disease. And in the schizophrenia instance, it's a genetic mutation or environmental insult that might be the driving factor. And in our case, instead of giving animal drugs of abuse, can I just rewire the brain of a mouse that had never experienced drugs and recapitulate addiction, if that's even possible? And predictive validity is that interventions on the animals predict a similar response in humans. And in this case, especially since I have this neuroeconomic framework that is translated across species, is what I'm doing in the rodent model predictive of what might happen to humans, particularly human patients. And now we have a framework to test them across species, these different interventions. And I want to make a point here about the difference between an animal model of a disease versus an animal model for a disease, where I'm not necessarily trying to model addiction per se. These mice are running around shopping for food. They're not giving themselves drugs. But I am using this as a model for studying addiction because it tells me something about information processing in the brain. So with that in mind, I've worked with Dr. Kelvin Lim and Jasmine Kamchong to collect preliminary data in human substance use disorder patients on this task. And so far we've collected in cocaine patients and healthy match controls. And what we find is, if I just cut to the chase on this interesting metric that we can now operationalize between what you should and shouldn't do. On the x-axis here, this is your probability of turning down an offer that's above your threshold. This is something that you should do. You should turn down bad deals. And alternatively, you should accept good deals. And what we find was the cocaine-dependent individuals, showed this impairment where they're essentially making decisions at chance when given a bad deal, which kind of looked a lot similar to our cocaine-treated mice. That cocaine-treated mice showed impairments when making the right decision, particularly for offers they should walk away from. And we did not see that in our morphine mice at all, even though they spent more time deliberating about those decisions. So if you think about these different neuroeconomic frameworks like foraging versus deliberation, it seems to be the case that cocaine-treated mice and cocaine-exposed humans share a similar vulnerability in a deliberative model that might not necessarily be true in a foraging model. And we don't yet have data collected in morphine-treated humans, but can we dissociate different types of patients on this task that we know are rooted in different circuit dysfunctions? So with that in mind, this has been work specifically in substance use, and I would like to think that this can kind of expand more broadly than that. There's been a number of individuals that have been translating these efforts across species for substance use disorder, but the laboratory of Dr. Hanos and other colleagues are moving this into anorexia nervosa. I've been working with other folks in the animal realm, particularly with Sylvain Linné's lab and Rachel Anderson, of bringing genetic mouse lines of disorders into this neuroeconomic framework, where they're wrapping up a study looking at aging, sex differences, and Alzheimer's disease on this neuroeconomic task. And there's other folks in the human realm looking at neuromodulation, whether it's recording neural activity, using functional imaging, or invasive or non-invasive neuromodulation approaches. And what we're really excited about is now Ben Hayden and his group are bringing monkeys into the picture to really get this cross-species pipeline in the works. And what this looks like is keeping things naturalistic to the animal. Where can you have a monkey foraging in a cage in different restaurants? And he so graciously provided this video of a monkey barring and swinging from restaurant to restaurant, essentially choosing between offers that might be expensive or not.
Good. Oh. Oh.
So whether or not we can get to this stage of a monkey surfing on social media to make these same types of decisions and have the same monkey do the rodent version of the task and something that we'd like to see humans happens, who knows, it might be possible. Right? But it's very clear that something very special is happening here at the University of Minnesota, where we can think about disorders that are transdiagnostic or move beyond classic boundaries of diagnoses and into this realm of computational processes that might be shared between different types of patients or might be different between different types of patients. So I want to just end on this note of impulsivity and self-control, particularly on the spectrum of development. I think everyone's familiar with the marshmallow task in kids, and that behavior on this task may or may not predict behaviors in adulthood, whether or not that might mean likelihood to fall to substance use or other forms of delinquency. And clearly, if I've got one thing across to you guys today, it's that impulsivity is a loaded term and it can mean a lot of different things. Particularly how the brain might be changing over development, might give rise to different vulnerabilities or different components of resiliency that either might strengthen or weaken over the course of development. And we really ought to think about the circuits that are changing underlying these different decision-making components. And so this little monkey that I have playing on this game is my niece before she was really verbal. And even she could search and surf the web on YouTube between videos that she wanted to use and didn't want to use. And I feel like naturalistic tasks like these can get at behaviors before an individual can even communicate those behaviors to us verbally. So I'm going to end there. Just to summarize, I've talked to you a little bit about the differences between how neuromodulation for different types of drugs of abuse could be fundamentally different. And really, if you can explore your data space, it can reveal a lot about the different subjective valuation systems that might not be obviously measurable using simple behaviors, and that we can target some of these pathways using new neuromodulation interventions. that we've translated this across species, and then now that we're moving to really resolve heterogeneity among different types of patients. So with that, I'd like to thank my 2 thesis advisors, as well as the number of collaborators that I've worked with over the years, my funding sources, including the translational funding grant I recently got to move a lot of this preclinical work into human patients. So with that, I'll take questions, and really thank you guys for your attention.
Brian, so awesome. Thank you. Question, did people's decisions change over time? So if the rodent was hungry initially, and then were they more likely to reject a long wait? Or if the humans started out excited about surfing, but then at the end were kind of bored of cat videos? Because we all think of things like things that make someone make a poor decision, hungry, angry, lonely, tired. Did any of that play in?
Yeah, so the short answer to that on the rodent side of things, because of course, right, satiety, they're getting full. If you just split the hour session in half and look at the first half to the second half, in rodents, it wasn't obvious that behaviors were changing. They weren't even slowing down. And I know that Dr. Hanos has found some interesting data as far as how behaviors might change over the course of that session and what that might mean for satiety or habit learning or other things like that that actually might be different between healthy individuals and individuals with anorexia, for example. Feel free to comment if you want. But there's a lot of interesting questions there about that. On the other side of it, if I purposely feed animals ahead of time and just like load them full with food, right? There's a certain point where I can start to see effects like that, where now they're going into the task for that day sated a bit. And the interesting thing is it depends on what flavor I feed them with. If it's something they care about versus something they don't care about, completely different things happen on the task that can be separated from satiety alone, right? Which is adding a whole other level of complexity to what the brain is responding to or not. Or I can at least remove the chocolate cake component out of the rest of what might be going on.
Excellent talk. Yeah, really, really interesting. I had a quick question with regards to the clinical study that you're doing. Were they cocaine-dependent patients? Or are they actively in recovery? Or
so that initial pilot study, which was done 3 years or so ago, wasn't selecting for what state of dependency was. Could some people even have been on drugs when they were on the task versus been in acute withdrawal? That was a kind of a first shotgun approach to just get initial preliminary data. And those were patients that were, those were subjects that were recruited from the internet. Right?
Interesting to see
how
their data changes during the recovery process.
So the current, the IRB for this is currently being under review right now. And we're recruiting patients from Lodging Plus to the Fairview system here. And we know these are patients that come in, they get tested routinely after acute toxicity withdrawal, and I'm learning more about how they get cared for in this particular facility. The majority of these are opioid-dependent users that might be starting methadone or transitioning to other types of therapies concurrent, and that we're interested in looking at multiple time points and looking at individuals having either saliva or urine drug screens throughout that process, as well as looking after they get discharged or after they leave this facility for follow-up time points as well. Absolutely.
Hi, Brian, great talk. You mentioned briefly that impulsivity is such like a loaded term, very multidimensional. And one facet of impulsivity that I'm very interested in is urgency or negative urgency, the idea that impulsive choices are more likely to be made under acute stress. And I know that you mentioned that acute stress could, as a trigger, reinstate addiction or addiction-seeking behavior, drug-seeking behavior. Yeah. I was wondering if you have any data or ideas about this concept of acute stress in the restaurant row task, and if that would affect errors of perseveration, like waiting, or errors of impulsive decision making.
Yeah, those are really good questions. So the fact that we have this framework now, all of these experimental questions can be, like, we can tackle these things now, right? It's just a matter of doing these experiments because we have the framework for it. So specifically within the restaurant row task, what I showed you as far as related to the drug data, I showed you individuals after prolonged abstinence, right? Kind of right before we would trigger them to relapse, whether it would be drug again or stress or something like that. If you recall from the condition place preference task I showed in the beginning, you can use stress or drug as a trigger for relapse. And so in that paper, in Nature Communications, we re-exposed animals to drug again, right, as a model of relapse, right? Not during the task, but we just gave them a reminder of what that drug was like and looked at how changes in what might be going on during that state that in those simple tasks have been used to model relapse, how that affects economic behavior on this task. And it actually looks different depending on their drug use history and what that acute event does. Now, we didn't do stress, and that's the next thing that's kind of on the list, is that if you take individuals that have been abstinent for some time, they're already in a decision vulnerable state, depending if they were cocaine exposed versus morphine exposed. And then you expose both of those to stress, which we know can trigger relapse-like behaviors in simple animal models. What does that do to decision-making vulnerabilities at that point? I did run an initial preliminary pilot study on exposing animals to stress on this task, and it was far after the entire experiment had taken place that they had gone through so much. It's really tough to make sense of what was actually going on. We need a separate experiment just looking at animals treated with stress on this test to really tease those apart. So the next question is, what's your intervention or what's your experimental manipulation to stress these animals out? And that whole literature of stress, that was the first type of lab I worked in, was in a neuroendocrinology lab, is your model of stress now can open up a whole bag of worms, whether it's physically restraining the animal, exposing them to a chemical stressor, or pairing the animal with a bigger animal that beats up on it. And this psychosocial stress can look similar in some regards to a physical stressor versus very different in some regards. And the fact that these animals are chronically food deprived and that they're working to sustain their own food livelihood is kind of this chronic stress imposed on this entire framework to begin with. So yeah, that's another whole loaded statement. But it's super interesting. There's a bunch of questions you can kind of go from here.
Thank you, Brian, for a beautiful talk with beautiful slides. So you brought up the marshmallow task, and my question is around that because it's recently failed to replicate. And the current interpretation is that what really drives those long-term findings, so impulsivity in the moment, was kids who are more impoverished, so socioeconomic status. So kids who are more impoverished are more likely to go for the marshmallow because they're living in a deprived state. And then because of that, that's probably driving their longer term outcomes. So my question is, as we move these tasks to people, how do we contextualize it within a person's broader psychosocial, environmental context so we're understanding that interplay between brain and environment?
Yeah. I think there's maybe 3 concrete ways to go about it. Of course, there's every patient, every human subject that comes through this task, knowing their past history and their environmental background can play a huge role in explaining individual differences on performance on this task, whether they come from an affluent background or a more impoverished background. So that's kind of actually taking in their environmental state, right, their baseline. The other thing is the reward that we're working for here, right? So working for food to sustain these animals' life, in a sense, versus entertainment and luxury items, also is another thing that you can play with if you change the prize item that they're working for. And whether or not some of these reward items might be triggering for different individuals. If we change from entertaining videos on the internet to emotionally charged images or videos or things like that. The other thing that I have some data on is the economic environment of rewards on the task. Right, so there's 3 different aspects of environmental richness. And I kind of skipped over this pretty quickly, but I'll just go back to it, particularly the mouse data. So going back to the sunk cost fallacy as far as irrational behaviors across training, what I showed you was the data over here of a well-trained animal who kind of knows how to thrive in a reward-scarce environment. And if you back up behavior to the first time they entered into a reward-scarce environment, their behaviors, their sunk cost behaviors in particular, look a little different. The graphs don't look much different at a glance, but if you look at all the colors of these lines, they're all rotated inward a bit, right? And so what does that mean? And the line I'm particularly interested in is this blue line versus the blue line over here, right? It seems like after waiting 1 second in the weight zone for animals is no different from the black line. So the green and black line are on top of each other. And the blue line, after waiting 5 seconds, is when you start to see that irrational inflation of value start to pick up. We don't see that happening in animals in a reward-scarce environment that they've had no experience in. But it does start to take off after 5 seconds. And so what that tells us is what's going on in the brain where those first 5 seconds aren't being factored into sunk costs, where they are later. And to me, that tells me that what decision process they're mentally engaged in is different, right? That those first 5 seconds, they're still not quite dwelling on the past like they might be here, right? And so if you start to play with these very sensitive variables, this tells us that the first 5 seconds, whatever the animal is doing during the weight zone, they're somehow not thinking about past actions, and they still might be thinking about the future or ignoring that information. And if you back these animals up to a more reward-rich environment, the sunk cost effect goes away entirely, right? And that being in a stressed nature, the nature of the environment being taxing on the animal might change whether or not behaviors are irrational or not to begin with. And I know individuals in child psychology that have studied the sunk cost effect in children and whether or not that goes on to predict delinquency in adulthood show results all over the place, that it either predicts susceptibility to delinquency or it's protective in some cases, or there's no effect. And I think the real question is, what's the neural substrates that are governing behaviors in each of those cases, whether it's stages of development or environmental richness?
Two related questions. When you are looking at the behavior in general between the cocaine and the opiates, is the speed of locomotion or the number of ultimate visits to the different restaurant the same, or they're higher with the cocaine just out of the rather excitation kind of element?
Yeah, I skipped over this a bit, but if you count the number of laps that these animals ran, how many pellets they earned, their body weight change before and after every session, and their speed in between every restaurant, those all look the same between groups.
That's pretty remarkable in its own right. But so the follow-up question to this.
And one point of importance is, remember, this is behavior at prolonged abstinence 2 weeks after their last dose. In the paper, in the supplement, we show you while they were acutely in withdrawal or after a relapse dose. And you might see some speed change differences when drug is either on board or just recently clearing. But again, I'm interested in a time point that might be relevant to this relapse-like behavior.
Right, but I mean, once they went and got cocaine, now they have cocaine in them, no? Or
so during this period,
yeah,
when they were exposed to drug, we gave them injections of cocaine at like 8 p.m. and we test them every day in the morning. So in theory, it should at least clear their system before the next time we test them. But the behavior I showed you was at least 2 weeks after their last injection.
And the follow-up question, which I know this is a memory and learning kind of a cognitive neuroscience approach to understanding the data, but I was struck with the time spent in society, right? The time after consuming and kind of staying longer there. And you know, whether pleasure as an affective experience is a modulator to those kind of circuit in a way, and whether that, because typically society is more endorphin opiate related, whether that kind of lengthening is actually starting to tie in with how the decision making about the opiate versus the cocaine is this time differential aspect.
Yeah, maybe. So are you asking if, and if I heard you right, society? Or can you explain that last part about the pleasure aspect of things?
You talk about the savoring phase where they seem to take a break after and kind of just hang out and savor what they just did. Was that working?
Yeah, let me go back. I didn't think there were. So this particular behavior, this lingering behavior? Yeah. Yeah, again, that wasn't different.
No, I know it's
right.
But I think that is typically associated with endorphins, right? I mean, so with the opiate system in general, rather than a dopamine kind of a thing. So the seeking would be a dopaminergic activation, and the savoring, the society, that pleasure you get after consuming something you really wanted, you take the time to savor it, is opiate. And that time is also an influence on the decision making in a way.
Yeah, I 100%
And it seemed
agree.
to be following more the narrative of your morphine rats than it does the cocaine seeking.
Right. So, okay. So thanks for clarifying that. So now we're getting into, I guess, a different nomenclature for these things that have been used in neuroscience by Kent Berridge and a few others, is this difference between wanting and liking, right? And that there seems to be an instrumental goal-oriented component of wanting, right? The fact that if you want something, you're trying to obtain a particular thing, versus liking, which is this kind of savoring component. And there's a long-standing animal neuroscience literature that these live in different circuits, right? And that hedonic pleasure is a different computational process than wanting or goal-seeking behavior. And so if I were to, in our task, use that nomenclature on these behaviors, I have maybe 3 distinct types of sets of behaviors. There's decisions in the offer zone, and there's decisions in the wait zone, both of which are in pursuit of a goal. So I would categorize both of those under wanting. And then there's this post-consumption process that I would categorize under liking. There's no overt reward, right? So in the data that I show you right here, there's no behavioral change in this liking component or this post-consumption process. But there was differences in 2 flavors of wanting, right? Depending on the drug of abuse the animal was exposed to. Again, also in this paper though, when we re-expose animals to drugs one more time, in both cocaine and morphine animals, we see a change in this post-consumption hedonic valuation. So again, I think we're accessing 3 completely different valuation processes here. And some that are deliberative in nature, some that are foraging in nature, that might be more retrospective looking, and something that is entirely different than both of those. And then they change on different timescales.
Thanks for the great talk. So my question is about your human research and the task in and of itself. And do you have insight into the circuits that are engaged, or even the brain components that are engaged during this task? Because if we can understand how these circuits are engaged, then maybe we can use this as some type of diagnostic criteria when patients come into the hospital, or even for the research groups that are combining it with non-invasive neuromodulation. Can we use this behavior to engage specific circuits to kind of modulate the neural mechanisms that we're looking at?
Yeah, I think that opens up a really good point. I want to go back to this slide as far as different ways to interrogate or probe these circuits. So what I showed you was by manipulating the strength of a circuit, of a very specific circuit, and then letting the animal behave for the next week, I can augment one very specific type of decision-making behaviors. Right? And so you could conclude from that 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 circuit is at least critically important in that behavioral process. Or at least the strength of that circuit is, the weight of that circuit. To be honest, I don't know if I were to go in this type of an approach and stimulate that exact same circuit during behavior, if I'd see, I don't know what I would see. I could turn on that circuit, I could inhibit it, and I don't know exactly what the brain is doing during this process versus this process in that particular circuit in the moment, right? It very well may be that if I manipulate that circuit in real time during this type of a behavior, I might see an effect. Because what I'm doing in that case is I'm imposing some unnatural pattern in neural activity that the rest of the brain has to work around in that particular moment. So if anything, just thinking about what your measurement is and what you're going to do if neuromodulation becomes something you want to now apply in patients on this task, is the goal to interfere with ongoing information processing? Or is the goal to affect a mechanism of learning and memory that might not necessarily translate to what the brain is doing in that moment?
Yeah, I guess it would be really interesting if we could use some type of behavioral task to really understand what brain circuits are involved. Can we use behavioral tasks to understand?
a good point.
Question. Brian, thank you so much. This is just fascinating. And the application, the translation to humans is going to, as you know, is going to be really fascinating. So a couple thoughts from the clinical realm to consider. We know that there's different, with different substances, there are different courses. So both in terms of development, initiation, maintenance, and reinstatement, and the pathways for each. And the time course, I think, is also different for different substances of abuse. And we know that relapse rates in general are highest in the first 6 months. So kind of examining, why is it some people more normalized sooner than others, or with certain substances or not? So those are just all kind of considerations in the translational world.
And I'm so glad that you're my mentor on that grant, that we're going to work on this together.
Thanks, Brian. This was beautiful. So kind of zooming out and looking at the bigger picture that you talked about, kind of seeing addiction as and other sort of symptoms as proxies for deeper causal disease states. How do you see this picture developing as far as like the goal moving forward? Is it to taxonomize different decision-making algorithms? And can this realistically be reduced down to a circuit level?
So ultimately, right, like what's the goal? And Amber, if I'm getting at your question, at what point is explaining all of this one goal in and of itself as much as it is to maybe develop new treatments or maybe new diagnoses? And I want to maybe just, we can talk all day about this, but I want to talk maybe just about that last part as far as treatment interventions go. The story about condition place preference I opened with, to me, was just like struck me as a cautionary tale, right? Especially with neuromodulation taking off as a new potential treatment avenue. And here's something that works brilliantly for addiction. And let's use this as a treatment therapy for another individual. Then now we're getting into a dicey territory of something that's disease preventing on one hand, and that same intervention might be disease provoking on another individual. Not just that it has no therapeutic efficacy, but that you're actually pushing an individual into relapse when they otherwise wouldn't. And part of it is largely due to we don't know what these circuits are doing in the context of the broader network. And for me, that's maybe just the one answer. I'll leave it at that. Is the goal for me is to understand what the brain's normally doing so that we can start to begin to intervene in a way that's meaningful and that's safest.
Hey, Brian, nice talk. My question is sort of off of the last thing you were just talking about with the clinical translation. I was going to ask, what are the next steps where you see this is going for treatment? And I guess to just elaborate a bit more on what you just said, do you think that there are therapy or cognitive training type therapies where you could train people who are addicted to substance uses or addicted to substances in order to learn how to make better decisions using this kind of paradigm where you have them make those decisions and investigate that?
Yeah, I think my response to that reminds me of stuff that David has worked on in the past, as well as some of Sophia's current endeavors on cognitive training. Right? We can talk about neuromodulation and invasive procedures all we want, but also there's a lot of utility into the power and strength of training. And it doesn't necessarily mean training on this task, as Dr. Vinogradov does in her work. Can we train up a particular circuit, something unrelated to, say, face recognition that might be an auditory task? And does that actually rewire augment circuits that are measurable on this task and that have implications for behavioral dysfunction for those types of patients? So I think of this as a two-pronged approach where I can categorize the behavioral dysfunctions on this task in a much more sophisticated way that's highly parameterized. But then maybe I can superimpose on top of that different training regimens that can augment behaviors measurable on this task in a way that's meaningful for recovery. And going back to some of David's work on contingency management, or individuals that respond to one type of behavioral therapy, like if you stay off drugs for a certain amount of time, you get a reward. And that driver, that motivator, works for some patients and not for others. And part of it is dependent on what decision-making system they're engaged in that makes them receptive to such treatment. And if someone's in a particular situation where they're not sensitive to contingency management, that might be trainable to move them into a certain circumstance where now behavioral therapy can work. Can we at least come at this from a neuroeconomic approach to measure that and monitor it?
But for the video people. Hi, Brian. Great talk. I'm an anthropologist, so I think about monkey behavior all the time. And the one thing that I was thinking about, like listening to your talk and seeing these slides, is a particular certain primate social groups experience transfer, where you have a male of a particular, especially in old world monkeys such as baboons and macaques, it's a social system in which the hierarchy among the females is more important than the males. So what you see is that the males are the ones that are going to be transferring between groups to achieve reproductive success, to essentially avoid incest within their own group. And what primate behavior has observed is that the lower ranking individuals are typically more successful in transferring between groups because they're able to successfully enter a different group as a lower ranking and therefore less intimidating presence in this other group. Whereas the higher ranking males in the original group are less successful in making that transition because they're not really used to being acting like a low ranking individual. And I'm thinking about this in the context of sort of this position of a behavior that wasn't necessarily negative to begin with, or sort of like an expectation that wasn't necessarily negative to begin with, but a situation that was changed in which all of a sudden that behavior was detrimental to the health of that individual. And I wonder if your research and if your experiments could sort of like speak to that as thinking about sort of like the mechanics behind that, but also if you can think of an application in modern human research, behavioral work, where certain behaviors may not be necessarily considered to be negative, but a radical change of environment occurs that forces the individual to deal with that change in a productive way.
Yeah, so Rutger, you bring up a couple of interesting points, one of which talks about how do you define what's optimal behavior in a given circumstance, and then the other aspect of this social environment. And I would love to talk to you more about this. Maybe I'll just mention one thing that might be interesting. And this reminds me of work from Michael Nader, who does addiction, substance use disorder research in monkeys, particularly in monkeys in social hierarchies. And if you take 4 monkeys and you put them in a cage, one will assert dominance, and you can rank the monkeys from top ranking monkey to the intermediate monkeys to the most subordinate monkey. And in monkey research, it's kind of hard to do. Usually you kind of have 1 or 2 monkeys trained in a long behavioral task and then really trying to understand the social dynamics and stress that might be involved in the social hierarchies. He's one of the few groups that does this. And what he found is in substance use, if you take that subordinate monkey and you put them in a task where they self-administer drugs, you see this dramatic increase in drug-taking behavior as a result of having morphed from a neutral monkey into a subordinate role, right? And that is a really interesting social framework to consider, but then the task is relatively straightforward. It's the animal self-administering drug. And I'd be really interested to see how complex behaviors, given that social history, can influence tasks or behaviors on an economic task where we can start to operationalize what's optimal and suboptimal. I can't really speak to what's optimal and suboptimal behavior in nature as far as sexual fitness or mating behaviors that might happen, but at least that there's ongoing research about the intricacies of dominant-subordinate social structures, particularly in primates, non-human primates, and how that leads to maladaptive behaviors in substance use. And so I'm absolutely interested in crossing those boundaries, but maybe I'll leave it at that for now.
Well, I'm aware of the time. I think we could go on for a couple more hours. I want to thank you again, Brian, for a real tour de force. I feel like we should give you a standing ovation. It was an excellent talk and a great discussion. Thank you, everyone.
Thank you.
You have been disconnected from the meeting.